Hello, everyone, and welcome to today's webinar titled Development and Manufacturing Strategy for DNA-Based Drug Vaccine Candidates. This webinar is part of the 5th annual event in the Bioprocessing Virtual Event Series. I'm Shelley Mulock of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to genscript.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located to the right of the abstract. This survey will be available during both the live session and the on-demand period are displayed. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby credits. I now present today's speaker, Dr. Mark Wei, Senior Director of Plasmid and Virus Process Development and Manufacturing Department, GenScript PROBIO. And Dr. Wei will be assisted by Hui-Yi Zhu. For a complete biography on our speakers, please visit the Presenter tab from the menu at the left of your screen. Dr. Wei, you may now begin your presentation.
Hello, everyone. So today we're going to present for development and manufacturing strategy for pDNA-based drug vaccine candidates. So before we start, I would like to introduce our company. So we are PROBIO, a global CDMO company nurtured by GenScript. Our manufacturing footprint and sales footprint expands in China, Korea, Japan, EU, and US market. We currently have over 300 plasmid and viral vector projects undergoing a different stage from CMC to commercial manufacturing. And as a global company, we have formed partnership with numerous leading organizations across the world. Our plasmid has been utilized by over 160 big pharmas and top biotech companies worldwide. So first, I would like to introduce some of the pDNA-based vaccine drug marketing and trending. So the outlook of the DNA vaccine market appears very promising. Currently, there are no available candidates for human use, and only 3 options exist for animal use. This leaves significant room for the development of new products. As of 2022, more than 57% of the DNA vaccine projects remain in preclinical stage, while the remaining 43% are undergoing clinical trials that necessitate the use of GMP plasmid manufacturing. Of DNA vaccine projects, nearly 20% of those are focused on COVID-19, which of course has been the major topic of the discussion in the recent years. So plasmids are of the most importance in the development of advanced therapeutics. It serves as a key raw material in the manufacturing of LVV or AAV for cell and gene therapy drugs. Well, it also serves as a pDNA vaccine drug product. And today, that's what we are going to focus on. So for the plasmid development and manufacturing strategy is what we're going to discuss next. So what is plasmid? So plasmid is a small circular DNA molecules in the cytoplasm of the cells that can replicate and transfer autonomously, and often carries a gene information that is beneficial for the survival of the host cell. So in the nature, the plasmid exhibits virus shapes under the electron microscope. So over here, we can see different shapes that the plasmid in the nature presents. However, during our production, our goal is to obtain the supercoiled plasmid as our final product. As for the DNA vaccine, the mechanism is shown in this picture. It uses a genetic material from a pathogen to stimulate an immune response. It contains a plasmid that has the inserted gene once the plasmid completed the transcription inside the body, it will generate mRNA, which will further express antigen inside the body. So to carry out this genetic material, we can use a DNA plasmid, a microcircuit DNA, or a Dodge bond. A Dodge bond is a DNA structure, also known as a dbDNA, that is modified DNA molecule, typically using in the preparation of a gene therapy or a DNA vaccine in this case. A name derived from its shape, which resembles a dog bone. So the FDA has two important approval stages for drug development. The first being the IND investigational new drug stage. It evaluates all drugs that are ready for clinical trials for the laboratory use. And next is the new drug application stage, which evaluates all animal and human data to approve drugs for treatment of disease. So over here, for the pDNA drug development, the roadmap typically started with a basic research, followed by a CMC, chemistry, manufacturing and controls, and non-clinical testings. After that, the IND submission is made, and 3 phases of study is conducted, including phase 1 for safety evaluation, phase 2 for dose efficiency assessment, and phase 3 for efficient comparison to the current therapy. The development of plasmid drug involves several key stages, including cell line development, process development, formulation development, analytical method development and validation, and scale up and GMP manufacturing. So first, a high titer stable cell line is selected, and a master cell bank is then established. Then, process development stage will begin with a small-scale process to identify critical process parameters and quality attributes. A formulation development then is made for plasmid, which is pretty typical for a straightforward process due to the stability of the material, but do need additional investigation for this is due to the plasmid was made into final drug substance as the vaccine itself. Analytical method development and validation is crucial to control batch release of the final product, which finally, after the process has been optimized and validated, it can be scaled up for pilot scale drug product production, and then GMP manufacturing, followed by stability studies. In order to provide a detailed account of production process, we'll begin by outlining those steps. The initial and crucial step involves the selection of suitable bacteria strain for the cell banking production. As the function of the different strains can vary during the cell culture stage, this will illustrate a family map of those different distinct strains in the bioprocess selections. Understanding those virus genotypes of E. coli and their associated phenotypes can aid in identifying an ideal strain. Ultimately, this will lead to a significant improvement in plasmid manufacturing process. Gene knock-in and knockout method can be employed to modify those strains to meet specific process requirements during our development stage. So a proper sourcing of the host strain and template plasmid is vital to kickstart the pDNA CMC study. At PROBIO, we ensure a clear history of the strain used for plasmid manufacturing and guarantee that these strains are licensed for commercial use with sub-license authority to prevent any roadblocks during the late-stage GMP manufacturing process. Additionally, we also offer support throughout the BLA and DA process and future commercialization efforts. For the cells, we provide the original records for strains and development records. We also provide the gene synthetic records for the plasmid constructions. So over here, we'd like to show some of the case studies that we have. This case study demonstrates the importance of a strain screening and selecting a strain with a good passage stability for cell banking is critical. So a poor passage stability strain was found to exhibit an extra band at the 8th passage, and this could be because of the same origin conflict. The extra band become more even pronounced at the 10th passage. This extra band material will be difficult to remove during the downstream processing. On the other hand, The string B over here shows no extra band even at the 10th passage. Furthermore, those second string exhibits a great stability with a high plasmid yield and the retention rate, make it more suitable for a large-scale production as no plasmid would be lost during the scaling up process using this good string. So here we demonstrate a process overview for our plasmid manufacturing process, this including upstream and downstream processing. So we start with the fermentation, then we collect the cell for cell harvesting, we lyse our cells, and through clarification process, we further down to concentrating of our material and moving on to downstream processing. So we first do geofiltration, which removes our RNA. Then we move on to affinity columns, which capture the supercoiled DNA, and move on to an AEX process for polishing the material. Further down, we do an UFDF again to exchange out the buffer for our formulation during the process. So our own in-house strain offers a full traceability and sublicense authorization, and our suspension culture is free of animal origin and antibiotics. Our high-density fermentation process is in upstream has a yield up to 1 gram per liter. This resulting in a cost-effective product with a high yield. Additionally, our downstream process bring a recovery between 20 to 60%, depending on the product specification. So this is a path for IND and BLA CMC study. We started with a preliminary evaluation, then follow up with the process development and a small batch production. To that, we can lock down the critical process parameters and move on to process evaluation report and settle on the process specifications. An IND batch production then is completed, followed by a process characterization and validation studies. Finally, the BLA will be submitted as is. So asset development is a crucial component of our process development for plasmid manufacturing, as it forms a key part of our risk-based analyzed control strategy. Through this strategy, we aim to ensure the robustness of our manufacturing process, with the right level of process control to mitigate risk and maintain the quality of our process, from raw material throughout to final product. So we also took a very systemic approach with the optimization process during the fermentation stage. So by evaluation of all of those key factors of each project, we determine the perfect condition for each process in order to increase efficiency and improve product quality. Those key factors could be substrate, which is the feed to the cells, pressures, stir speed, airflow, viscosity, and osmotic pressures. Here's a picture of our upstream manufacturing process equipment that we use on site in PROBIO. So experimental design rely on fundamental principle of fermentation. This include energy utilization, metabolism, and information transfer. We choose fermentation process aligned with the plasmid sequence and the bacterial growth rate, which enable us to achieve a high yield and improve quality. After optimization, our fermentation yield can reach up to 1.5 gram per liter. So we would like to present a case study outlining the optimization of our upstream process. Specifically, we made adjustment to the pH level, components, and concentration, resulting in medium optimization of this process. And the outcome of this modification exceeded our expectation, with the yield increase by almost threefolds and surpassing the 1 gram per liter mark at the 32-hour mark. Additionally, our growth of our product is now more consistent and stable after optimization has been happened. So regarding the downstream platform of our plasmid, the process we utilize is highly effective at eliminating the impurities and contaminations, those including HCP, HCDNA, and endotoxin. This results in a high concentration of drug substance for production. Doing our most successful batch demonstrated a purity of over 99% of supercoiled plasmid and less than 0.1 for cDNA, and with the level of HCP falling below quantification limits. Our endotoxin content is measured less than 1 EU per milligram at our best performance. And here, we're pleased to present another case study highlighting the successful optimization of our client upstream and downstream process. In the upstream process, we made modification to the culture condition and supplement strategy. This results in an impressive 2.6-fold increase in optical density and 4.8-fold increase in yield. Similarly, during the downstream process, we were able to improve our ultrafiltration and diffiltration process recovery rate from 73% to 90%, while also reducing the whole cell DNA content from 0.3% to 0.1%. So here's another case study on the stability of a pilot scale production. Once we have established the process parameters, we conduct 3 consecutive runs with a pilot scale. Those data presented over here indicated a very high reliability of our manufacturing process. The OD-CV is less than or equal to 5.1%, and the yield CV is less or equal to 11% for the fermentation process. This demonstrates a very stable production capacity on the later stage process. So once all the upstream and downstream process has been concluded, we move on to the formulation process of our drug product process. The DP formulation development process involves 3 major stages. The first stage, the pre-formulation, consists of designing of the formula, conduct compatibility study, and predict the stability. In the second stage, formula development, we screen and select appropriate packaging material and complete stress and stability tests to assure our product can be stable during a harsh condition in the selected formula. Once all parameter has been optimized, we scale up the production and complete verification and process proceeding to the manufacturing for GMP filling. And with the new needle-free technology, delivering the method of the plasmid is very easy and effective. So moving on is to our quality system. So quality is an essential factor throughout the entire production process, starting from the vector design and continually through the release of the raw material in process control and product release specifications. It is throughout the whole manufacturing, quality is the key. So a range of tests is performed on the releasing of the raw materials. Those are conducted to ensure the production of a high-quality drug product from the beginning. This includes characterization of a cell bank and stability test, characterization of the strain bank and the stability test, release of all the raw materials, including release of all excipients and the packaging materials as well. To ensure the quality of the plasmid, a bunch of testing is also performed in 4 different categories, including physical and chemistry property, characterization, purity, and safety. Those different methods is available in the house for us to verify the product quality to ensure the plasmid is safe and stable. So in addition to all those various tests on plasmid, our method qualification is also carried out during determination of the acceptance criteria for the assays. Once this analytical method are qualified, those product material then will need to pass all those criteria so it can be released to the manufacturing team. Here's in a case study for overall seeing indicates a high quality plasmid product. First, we can see the HPLC analysis of the plasmid indicate that there's over 80% of the content which in form of supercoiled plasmid. And over here, the restriction digestion analysis matches the anticipated pattern, as well as the plasmid sequence analyzed also corresponding to the predicted DNA sequence, shows a high quality of the plasmid product. So we also utilize 3 distinct in-house methods to access the plasmid purity. This is tailored to specific need of each stage process. For example, agarose gel electrophoresis is fast technology to use during the cell banking stage. This ensures a fast turnaround time. And during the process development stage, an AEX HPLC is utilized to provide a more thorough analysis of the plasmid purity data. And finally, in the final project stage, the supercoiled percentage is typically accessed using a capillary electrophoresis method to obtain a high resolution data on the purity profile. So we also possess a cutting-edge sequencing technology in-house, which allows us to improve detection of certain genetic features. For example, with a traditional Sanger sequencing, detecting the inverted terminal repeats can be very challenging. However, our ITR sequence technology in-house can accurately read through ITRs, providing a clean peak that enhances our vector stability and safety. Furthermore, we have recently upgraded our poly-A sequencing capacity, which enable us to read poly-A up to 140 pairs and provide more comprehensive and detailed information about transcriptomes. Further on, the cost of the manufacturing process could be pretty high, but developing a cost-effective and sustainable manufacturing process requires careful consideration before launching the product. To this end, we follow a certain approach every single time when we're designing a new process, making sure the capacity, flexibility, scalability, cost, quality and regulatory, and speed in order to deliver a robust process to drive down the cost of goods sold of a plasmid. So through a systematic analysis of a manufacturing cost model and available production technologies, it is possible for us to identify options that can deliver the required level of productivity for a large-scale plasmid production while still meeting batch costs constraints and aligning with the reimbursement strategy. By doing so, it is possible to reduce the cost of goods sold, which is typically pretty high for a large-scale plasmid production. The Plasmid CDM platform for GeneScript PROBIO will be shown next. At PROBIO, we have over 2.5-acre manufacturing facility, which is ready currently, and 5-acre in constructions right now for plasmid manufacturing capacity, including CMC, cGMP Pro, and GMP capacities. This can be utilized for cell and gene therapy process development, mRNA, DNA, therapeutics, and vaccines, as well as the CRISPR gene editing techniques. We are also currently constructing a new state-of-art facility that is scheduling to be commissioned in the second half of 2024. This new building will provide a GMP operation capacity of approximately 2 acres. This facility will be also equipped with the latest technology, including Factory, and it will offer a range of service from upstream cell culture to automatic fill finish process. And importantly, this facility will comply with the current good manufacturing practice regulations, provide GMP capacity in US. So finally, I would like to demonstrate our excellent track record. So GeneScript PROBIO has an impressive track record over 20 global approached investigational new drug projects and more than 60 ongoing IND projects. We have also completed over 300 good manufacturing practice batches for clinical phase, covering a wide range of applications throughout cell therapy, gene therapy, and vaccines. As a technically strong vendor, we offer a range of advantage, including a ready-to-use lenti-helper plasmid for a third-generation LVV packaging for cell therapy. And we also provide a ready-to-use AAV plasmid for AAV packaging for gene therapy. We also have extensive experience with plasmid for mRNA with a poly A tail length ranging from 70 to 150 base pairs across more than 15 projects. In addition, we utilize cutting-edge technology for LcDNA with 3 projects currently underway. We offer very high concentration DNA vaccine for the effective applications. So on the right over here, we're proud to present those highlights for our IND-approved project, which listed right here. That will be concluding for the presentation today, and thank you all very much for your time. And we're anticipating any question and happy to help. Thank you.
Presentation. We will now start the Q&A portion of the webinar, assisted by Huai Zhu. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. So the first question that we have today is, what advantages does a DNA vaccine offer compared to other vaccine types, such as mRNA vaccines?
Oh, thanks for the question. So DNA vaccine itself has several advantages against mRNA. And first of all, it's very stable. So the DNA vaccine are more stable than mRNA vaccines as they are stored at normal refrigerator temperature without losing their effectiveness. And further on, the manufacturing of the DNA vaccine is also easier and cheaper compared to mRNA vaccines. Furthermore, I believe DNA vaccine is introducing a longer and stronger lasting period compared to mRNA vaccine, corresponding to the long immune response inside the body and triggers the T cell arms as well when they do. And additionally, DNA vaccine also provides a more safe condition compared to the mRNA vaccines on the other hand.
Okay. Oh, that's very helpful. Thank you so much. The next question that I see is, what is the typical turnaround time for an IND project?
So for that question, the IND project turnaround time is totally based on the project need. So usually, without any major modifications, the regular turnaround time should be around 8 weeks to 10 weeks.
Okay, very helpful. Next question is, do you provide any additional services to assist with IND filings?
That's of course. At GeneScript PROBIO, we provide end-to-end service for all the plasmid, including DNA vaccine services and cell therapy and gene therapy. So typically, we will provide regulatory affairs consultings, including any service needed for documentation preparation or response to FDAs, and additional to some clinical trial management. Also, we provide in-house toxicology testing, PK studies on the side if required, and further on, if the client is consider moving on forward to a GMP graded manufacturing process, we are kindly to provide the whole strategy and service later on. And also, very importantly, we provide all the quality control and the quality assurance services to ensure the filing of the IND is successful.
Oh, wonderful. Okay. Well, it looks like we have time for one more question today. And our last question today is, whom should we contact if we're interested in discussing our project? That's a good one to end on.
Oh, thanks for the question. So first of all, the easiest way is definitely to log in onto GeneScriptProbio.com that have all our service and informations listed on the internet website, and going through details for whatever service you needed it, then find the contact information later on to provide whatever information to our email or phone calls, so you can easily reach out to us, and our business development team will respond to you in the first thing they see it.
Perfect. Oh gosh, well, thank you again, Huai, for your assistance today. And do you have any final comments for the audience or thoughts?
I think I'm good.
Good. Okay, wonderful. Thank you again, Huai, for your help today. And thank you to Dr. Wei for your time and your important research as well. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsors, GenScript, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand for 2 years until April 5th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you very much. Appreciate it.
Thank you. Yeah.